Inside every single one of your cells, there's a book. It's the book of life, and it's written in a 4-letter code that spells out, well, everything about you. Today we're going to look at how scientists finally learned how to read it. And trust me, it wasn't easy.
I mean, think about the challenge here. How in the world do you read a microscopic book that's 3 billion letters long and get it perfect without a single typo? For decades, this seemed completely impossible. It was a total roadblock.
But the solution, when it finally came along, was just brilliant in its simplicity. The big idea? Don't even try to read the whole thing in one go.
No. Instead, you do something much cleverer. You make millions of copies of your DNA. But, and here's the trick, you sabotage the copying process. You force it to stop at every single possible letter along the way. So you end up with this massive library of fragments, a piece for every single position in the code. So the real question is, how do you pull off that clever sabotage?
Well, it all comes down to this really slick piece of molecular engineering. We call it the chain termination trick.
So think of it like this. DNA is being built on an assembly line, and the machine uses these normal building blocks, which have a special connector that lets you add the next piece. But what if you sneak in some faulty parts, special terminator blocks that are missing that connector? Well, when the machine grabs one of those, the whole assembly line just grinds to a halt. The chain is terminated.
Right, so in scientific terms, this special terminator block has a name. It's called a deoxynucleotide, or just ddNTP for short. That missing connector we talked about, it's a chemical group, the 3' hydroxyl group, and without it, the next DNA block literally has nothing to attach to. Game over. So to make this all work, you run the copying reaction with a mix of normal blocks and these special terminator blocks. And here's the other cool part. Each terminator is color-coded with a fluorescent tag. So A might be green, T is red, C is blue, and G is yellow. What you get at the end is this massive collection of DNA fragments of every possible length, and each one ends with a brightly colored tag that tells you exactly what its last letter is. Okay, great. So now you have this chaotic soup of billions of fluorescent DNA pieces, all different sizes, all just jumbled together. How on earth do you sort them perfectly from shortest to longest so you can actually read the message?
Easy. You stage a race. Welcome, everyone, to the Great Molecular Race.
Now this race has some pretty interesting rules. The racetrack itself is this gel, kind of like a really thick jello. The starting pistol, it's an electric current. And since DNA has a negative charge, it naturally wants to run towards the positive end of the track. But here is the absolute key. In this race, the only thing that determines your speed is your size. The shorter, lighter fragments can just weave through the gel way faster than the long, heavy ones. So, you load your DNA soup at the starting line. You flick on the electric current, and they're off. The fragments immediately start to sort themselves out. The shortest one, the 1-letter fragment, it crosses the finish line first. It's followed by the 2-letter fragment, then the 3-letter, and so on all the way to the longest piece. It is just an incredibly elegant way to create perfect order out of total chaos. So, the fragments are made, they're sorted. Now for the final and most important step. Reading the rainbow code. See, right at the finish line of our molecular racetrack, there's a detector. It's a laser that's set up to scan each and every fragment right as it finishes the race. So as each tiny fragment zips past, that laser makes its colorful tag glow. A computer sees that flash of light, records the color, let's say red, and then the next fragment comes through, and the next, and the next. And because they're arriving in perfect order of size, the computer can read the sequence just by recording the order of the colors, red, yellow, blue, green, yellow, which translates to T, G, C, A, G, just like that. This final readout, this beautiful graph you get at the end, is called a chromatogram. It's basically a visual representation showing the fluorescent peak for each letter, one after another, in perfect order. This right here is the decoded message, the printout from the Book of Life.
So let's just recap this amazing process. It's really just 3 simple steps. First, you terminate. You make millions of DNA copies that are forced to stop, creating fragments of every possible length. Second, you sort. You run all those fragments in a molecular race to organize them perfectly by size. And third, you read. A laser detects the color of each fragment as it crosses the finish line, revealing the DNA sequence 1 letter at a time.
And get this, This foundational method, the Sanger sequencing method, was developed all the way back in 1977. It was so revolutionary that it earned its inventor, Frederick Sanger, his second Nobel Prize. So why does all this matter so much? Why is being able to read this blueprint one of the most important scientific breakthroughs in, well, history?
It's because this technique was the key that unlocked pretty much everything else. This was the technology that was used to map the entire human genome for the very first time. It's what allows us to pinpoint those tiny genetic typos that cause diseases like cystic fibrosis or sickle cell anemia. It's how we can develop medicines that are targeted to a person's specific genetic code. And it's how we've been able to explore the vast tree of life, from the smallest bacteria to the biggest whales.
This one technique fundamentally changed our understanding of ourselves and the entire world around us. Which, leaves us with one final, pretty fascinating question. Now that we can read the book of life, what story will we choose to write next?